She went to the group of Javier Fernandez Ruiz in 2010 at the Universidad Complutense de Madrid. She has master's degrees in biochemistry, molecular biology, and biomedicine by the UCM, by the Complutense, as well as a PhD in 2018, also at the Complutense. She has been hired by Cybernet for several years, and in 2022, she got a position as ayudante doctor at the Universidad Complutense de Madrid. Throughout these years, she has been interested in developing new drugs, cannabinoid drugs, for the treatment of neurodegenerative diseases, especially for Parkinson disease, but also some others. And in the last few months, she has been able to get a grant from the Plan Nacional as an independent researcher, focused on the search for some orphan receptors role in Parkinson's disease. Her talk today is titled Preclinical Development of the Cannabigerol Derivative VCE 003.2 as a new neuroprotective agent in Parkinson's disease. So Maria, thank you very much for coming. Welcome to this webinar series. Thank you for your willingness to share with us your work. And the floor is yours, so you can start sharing the screen and go ahead. Thank you.
Is it okay, Miguel?
Yeah, I think
Can you
so.
see that? Okay,
Yep.
Okay, perfect.
All right.
I couldn't see anything. Thanks for the presentation, Miguel, and good afternoon, everybody. First of all, I would like to thank Cybernet and the organizers of this webinar for giving me the opportunity to present these results that we have obtained in relation to the preclinical development of the cannabigerol derivative VCE 003.2 as a new protective agent in Parkinson's disease. First of all, I will give you a brief introduction on how this molecule has been designed and synthesized, as well as its neuroprotective potential. Regarding the first point I have introduced you, there are different research studies that have demonstrated that many phytocannabinoids and their metabolites interact with the various PPR isoforms. But until the beginning of this research on these molecules, nothing was known about CBG and, of course, of its derivatives. It was for the first time in the year 2012 when our partners published this paper in which they explain an improved PPAR gamma activation capacity by CBG with respect to other phytocannabinoids that have been studied. Our partners also found that CBD kinone improved binding to this receptor compared to its natural cannabinoid CBD. Because of this, our partner decided to develop the first CBG kinone called VCE 003. As we can see in this picture, VCE 003 resulted a potential anti-inflammatory agent that increased the PPAR gamma binding activity of his natural cannabinoid CBD. We can see that the binding affinity of 003 was 2.2 micromolar versus the binding affinity of CBG, which was 12.7 micromolar. Our partners decided to develop a second generation of the cannabigerol kinone in order to improve cell viability and pharmacokinetic parameters, and so they obtained this molecule, the study molecule VCE 003.2, which I'm going to call from now compound 3.2. This molecule was evaluated through different assays to determine its potential as a future neuroprotective drug. Its binding capacity for PPAR gamma was first evaluated, and we found that compound 3.2 binds to the nuclear receptor with an IC50 value 1.2 micromolar versus the IC50 value 2.2 micromolar for the compound of reference. It was in the same range. The effect of PPAR gamma transcriptional activity was also measured for both molecules, and the transcriptional activity for PPAR gamma of both 3.2 and its precursor 003 was evaluated, as I say, and results showed that VC003 induced PPAR gamma transcription in a biphasic manner, and its activity was low with the highest concentration, as we can see that was 50 micromolar. On the contrary, compound 3.2 induced PPAR gamma transactivation in a concentration-dependent manner. In order to discard the mechanisms of action of this compound, the binding profiles of compound 3.2 for classic cannabinoids receptor CB1 and CB2 was also investigated by using ligand binding assays. This data confirmed that compound 3.2 has a poor binding affinity for both cannabinoid receptor types, with predicted K2B values higher than 40 micromolar. One more point of characterization of this compound consists in evaluate cell viability of this compound and its precursor using MTT assay in Na2 cells, which are neural cell line. We found that compound BCE003 showed a clear cytotoxic activity at the highest concentration tested, but it didn't occur with compound 3.2. Finally, in order to conclude this introduction and in order to characterize this compound, we wanted to characterize its neuroprotective potential by using several assays which include which evaluate three common mechanisms involved in the development of neurodegenerative disease, as excitotoxicity, oxidative stress, and neuroinflammation. In relation with cytotoxicity, we found that compound BCE003.2 was able to prevent this pathological event, which was induced by glutamate at a concentration of 15 micromolar in cells in N2A cells, and this compound was able to revert it, but compound 003 didn't do that. In relation to oxidative stress, while higher concentrations of compound BCE003 at 25 micromolar produce ROS liberation, release in this model, compound 3.2 didn't do that. Instead, compound 003 at this concentration produced ROS release at levels of control commonly used for its known and pro-oxidant activities, TBHP. Finally, in relation to the neuroinflammation, pathological event, compound 3.2 was able to attenuate LPS-induced release of TNF-alpha and reduce the p-values compared to controls in the case of interleukin-1 beta. This was measured in the media of culture BB2 cells exposed to LPS in the presence or absence of 5 micromolar of compound 3.2. Thus, at this point, the take-home message I want you regard to compound 3.2 is that the specific modification that our partners introduced in the structure of compound 3.2 first did not affect its paper gamma binding activity respect to its precursor compound 003. Second, this modification abolished its theophilic and cytotoxic activity. And finally, and more important, this modification could present a possible neuroprotective profile for this compound. Okay, as you know, at the neuropathological level of Parkinson's disease, the presence of intracellular aggregates of alpha-synuclein protein is highlighted. But in addition to this alteration in alpha-synuclein proteostasis, there are other underlying pathological events such as neuroinflammation, mitochondrial dysfunction, or oxidative stress. All of these interrelate synergically to jointly contribute to neurodegeneration. This multifactorial aspect of Parkinson's disease make it necessary to use multi-target strategies that can simultaneously reverse or attenuate several of these events. So given that compound 3.2 presenting, as I have told you several slides before, given that this compound presents this multifactorial role that include potential over several pathological events that occur in Parkinson's disease, we wonder if this molecule could have potential for the treatment of this disease. Thus, we decided to evaluate the neuroprotective profile of compound 3.2 in different models based on the reproduction of different pathological events that occur in Parkinson's disease. Well, I will first discuss the first model, the different models used to reproduce neuroinflammation, one of the pathological events that occurs in this pathology, and the results that we have obtained. We started these studies developing an in vitro model, which consists in investigating 3.2 capability to protect culture M213 neuronal cells against death induced by exposure to conditioned media generated from culture BB2, which were overexposed to LPS. Cell viability was measured in neurons by MTT, and these neurons were treated with different concentrations of compound 3.2 or with controls like rosiglitazone on in presence on or in absence of PPAR gamma inhibitor T0070907, which I call the rest of the presentation T0. We evaluate different concentrations of the static compounds, 0.1 micromolar, 0.5 micromolar, or 1 micromolar of BC003.2, and we use these controls as I have mentioned before. We found that a classical dual effect of this of cannabinoids by disease evaluation, in which at low concentrations of these compounds, 0.1 and 0.5 micromolar, neuron viability was preserved against the inflammatory insult as near control levels. And very near to group of the control rosiglitazone, who was used as a control, positive control for PPAR gamma, which I have told you. But at higher concentration, but it's not too many higher, at this concentration, we couldn't preserve cell viability. The three concentration tested and rosiglitazone were also examined in the presence of the T0 inhibitor PPAR gamma in order to determine whether this effect was mediated by the activation of PPAR gamma receptor. However, whereas the effect of rosiglitazone was completely reversed by T0, this inhibitor was inactive against the effect of the compound 3.2 at the anti-inflammatory doses. This suggests that the mechanisms of action of this compound could be other than the canonic activation of PPAR gamma in these effects. Because we have obtained protective effect of this compound, we wanted to explore if this molecule could have anti-inflammatory potential in a murine model consisting on a unilateral striatal stereotoxic injury with the pro-inflammatory toxin LPS. Animals were treated by intraperitoneal administration at a dose of 10 mg/kg for 21 days, alone or combined with the inhibitor that I have mentioned before. 24 hours after the last treatment, animals were sacrificed and their tissues were processed for analysis by immunohistochemistry and RT-qPCR. The results obtained for immunohistochemistry were as follows. First, we analyzed tyrosine hydroxylase, TH, from now, immunostaining in the substantia nigra pars compacta, and demonstrate a reduction of immunoreactivity in LPS lesioned mice. The reduction in immunoreactivity levels was reversed by the treatment with the compound 3.2. Then we wanted to analyze glial reactivity by measuring EVA1 and CD68 immunostaining in the substantia nigra. In both cases, we found a lowering effect of BCE3.2 in the intense microglias provoked by LPS. Such microgliosis was evident not only by the elevated immunoreactivity, but also about the presence of microglia cells leveled with EVA1 with amoeboid aspect, activated state, or compared with the ramified form, quiescent state, currently found in controls. We also analyzed by qPCR several pro-inflammatory markers typical of the activated microglia cells recruited at the lesion's ipsilateral striatum, always compared to the non-lesioned contralateral site. As expected, we detected a strong elevation in all the markers, that we have studied, related to the cytokines, TNF-alpha and interleukin-1 beta, as well in the pro-inflammatory enzyme INOS in the striatum of LPS lesioned mice. The elevation of INOS was attenuated by the treatment with compound 3.2, with differences between vehicle and treated LPS lesioned mice with compound 3.2. This was statistically significant compared with the controls. We didn't obtain this signification with cytokines, but we can see a very evident trend for the cytokines. Okay, seeing that promising results in this previous murine model, we decide to develop a more clinical approach by evaluating the inflammatory potential of these compounds in a murine model similar to previously described, but in this occasion, we treat animals by oral administration during 28 days. In this case, we also perform behavioral tests 2 times, one after the injury and the other just before the sacrifice. Also, we perform several behavioral tests with test rot-a-rot, actimer, and cylinder reading test. We only obtain significant data in this lastly test, cylinder reading test. So this is the data I'm going to show you. As you can see in this picture, our data demonstrate that LPS lesion caused preference for ipsilateral POE in the cylinder reader test, which was evident at both 1 week after the onset of the treatment and directly before autopsy after 4 weeks of treatment with the treatment with compounds 3.2 at the dose of 20 milligram kilo. We obtain a partial reduction, but the effect is very evident. Related to immunohistochemistry results, in this occasion, the histopathological analysis of basal ganglia revealed a trend towards a recovery by compound 3.2 at the dose of 20 milligram kilo, but not at 10 milligram kilo, as you can see in the picture, while we are measuring the loss of TH contained in nigrostriatal neurons caused by LPS lesion in animals. Regarding glial activity, also no differences were observed in microglia after treatment with the compounds at both doses, as you can see here. A partial reduction triggered by compound 3.2 at both doses in the intense axillary reactivity provoked by LPS was observed in substantia nigra, which was measured by GFP immunostaining. Lastly, in this model, we analyzed pro-inflammatory mediators expression by RT-qPCR, as I told you before. Results showed that the marked elevation provoked by LPS in general was attenuated by the treatment with compound 3.2, with the greatest effects found at the dose of 20 milligram kilo, except for this marker, interleukin-1 beta. Similar trends were found in relation with inducible nitric oxide synthesis, INOS, and cyclooxygenase COX-2, sorry. Also, the differences did not reach statistical significance in most cases. We think partially due by the high variability in the lesions groups, in particular in vehicle-treated LPS lesion mice. Below, I'm going to show you the different models and the results that we have obtained, different models that we use to reproduce this pathological event, which is very important in Parkinson's disease, which consists in oxidative stress. We started, like in the in neuroinflammation studies, with an in vitro model. In this case, we use SHSY5Y neuron cell line that were treated with the compound 3.2 at 4 different concentrations that were selected because of the IC50 value that we show in the first slides. 1 hour later, after treatment with the compound, cells were exposed to 6-hydroxydopamine toxin, and 24 hours later, cell viability was evaluated by MTTSA. Compound 3.2 showed cytoprotection in a concentration-dependent manner. We selected for next experiments the dose of 20 micromolar because the higher concentration, 40 micromolar, showed lower cytoprotective effect. In the next experiment, in where we use the selected concentration of compound 3.2, cells were treated at a similar similar than the experiment I have explained before, with the only difference that cells were pretreated with the inhibitor of PPAR gamma receptor GW9692 at the doses of 20 micromolar 30 minutes before the treatment with the compound. In this case, the objective was to determine if activation of PPAR gamma was the pathway of action of this compound, but we couldn't demonstrate that because the cytoprotective effect of compound 3.2 was not eliminated by the inhibitor GW. But because we have detected potential in oxidative stress treatment, we decided to analyze the potential of this compound in murine models for evaluate this potential at this level too. This model consists in unilateral striatal stereotactic injury with 6-hydroxydopamine toxin. Animals were treated by intraperitoneal administration, of compound 3.2 at a dose of 10 mg/kg during 7 days. Behavior tests were carried out 24 hours after the last treatment, and the animals were then sacrificed and their tissues were processed for analysis by immunohistochemistry. The behavior test was evaluated in this occasion by rotarod and actimeter, and we found that compound 3.2 significantly improved the motor impairment observed in lesion animals. This appeared as a mere trend in rotarod performance, but it was strongly evident in different motor parameters such as ambulatory activity, resting time, wearing behavior, mean velocity, and fast movement that was measured by actimeter. The histopathological analysis of the substantia nigra in these mice showed only a very modest attenuation in the loss of Th-positive neurons in the substantia nigra, which only consisted in a reduction of the probability level compared to control animals. In this model, we didn't observe changes neither by the lesion nor by treatment with compound 3.2 in glia markers, EVA1 and CD68. This result is concordant with the poor inflammatory response typical of this experimental model. Again, one more time, in order to make a more clinical approach, we decided to evaluate the antioxidant potential of compound 3.2 in a murine model, similar to previously described for neuroinflammation, even in Parkinson's disease, but at this time, the animals were treated orally for 14 days. In this case, the dose of compounds was 20 mg/kg. The behavior was evaluated in this case by pole test and cylinder reading test. The treatment with compound 3.2 caused an important improvement in the motor defects shown by these animals in pole test, reducing the time spent in descending the pole to evaluate similar to controls, as we can see here. The same happened with the hemiparesis shown by 6-hydroxydopamine lesion mice in the cylinder reading test, which was completely reversed by the treatment with compound 3.2, as you can see in this picture. Again, we studied similar parameters to test neuronal percent inversion and glial reactivity by immunohistochemistry. Compound 3.2 administration partially prevent Th-positive neural neurons against 6-hydroxydopamine insult, as reflected in the elevation in Th immunoreactivity measured in lesion substantia nigra after the treatment with this derivative. Although glial reactivity is not an important pathogenic event as we have described before, in this occasion, we detect differences that we can presumably assume as a secondary response caused by death of Th-positive neurons, a fact that we can find in this study with the results as I'm showing you. Because the effect produced this inflammation was completely reversed by the treatment with compound 3.2. The same response of 3.2 was also found when we study the astroglial marker GFAP. And finally, for this model, we also wanted to explore if difference occurred in the dopamine and its metabolite DOPAC levels after lesion with this toxin and after the treatment was the compound 3.2. With regard to the reduction in the concentrations of dopamine and its metabolite DOPAC observed in the striatum of mice lesion, this treatment with cannabinoid, with compound 3.2 elevate both dopamine and DOPAC levels in the case of DOPAC at the control levels, in the case of dopamine, more or less 50% of basal levels of dopamine. Finally, due to the important role of alpha-synuclein aggregation in Parkinson's disease, we decided to explore the neuroprotective potential of compound 3.2 in an alpha-synuclein aggregation murine model. Animals were unilaterally lesioned in the substantia nigra of the right hemisphere with inoculation of adeno-associated virus type 9 carrying the mutated synuclein gene. On the day following the lesion, animals started to be treated daily with this compound at the dose at 20 milligram kilo and during the next 2 weeks. Once the treatment was completed, the mice were subjected to the corresponding behavior test, and then they were sacrificed, the brain was removed, and the substantia nigra was used for analysis, immunohistochemistry analysis, and the striatum was employed for RNA sequencing techniques. In this case, the behavioral tests employed were cylinder reading test and elevated body swing test. In the cylinder test, we observed an increase in the preference for the ipsilateral paw in lesioned animals, which was completely reversed by the treatment with the compound. However, in the torsion test, we did not observe significant difference between the experimental groups. But we were able to establish a treatment effect when studying the general mobility in these mice, understood as the number of twists performed regardless of which way, thus observing greater mobility here, greater mobility in the treated animals than in the injured ones. Related to immunohistochemistry results, the virus injury produced a decrease of Th immunostained cells, which was completely reversed with the treatment with compound 3.2. Again, we wanted to evaluate glial reactivity levels, both in microglia and astrocytes. Treatment was able to attenuate glial reactivity, although it only was significantly in CD68, not in case of GFAP. Thus, taking into account all the models developed to evaluate the preclinical potential of compound 3.2 for the treatment of Parkinson's disease, we can conclude that in general, BCE003.2 prevents neuronal loss and decreases glial reactivity levels in Parkinson's disease in models employed. Compound 3.2 decreases several pathological events which occurs in Parkinson's disease, like neuroinflammation, oxidative stress, and alpha-synuclein aggregation. Compound 3.2 fully or partially recovers dopamine and DOPAC levels in the 6-hydroxydopamine model. This compound fully or partially recovers behavioral dysfunctions caused by animal models, and this compound could be a potential neuroprotective drug for treatment of patients with Parkinson's disease because of its multi-drug profile. And finally, I would not like to finish this webinar without telling you something about the future prospects for this compound. Of course, the objective of this on this drug discovery research is to obtain an approved drug for disease treatment. But in order to reach this goal, the potential drug must overcome several stages. We can say that we have completed a very satisfactory preclinical studies for this compound 3.2. We could think that we can pass to the next phase. And I'm also pleased to inform you that a phase 1 clinical trial for this compound has already started in this year in Australia. So let us, we hope that clinical research of this molecule will have promising results. Finally, I would like to thank my research group, which is led by our PI, Javier Fernández-Ruiz, and especially to Sonia Burgaz and Santiago Rodríguez for their enormous contribution to this research. I would like to thank all of your partners that have contributed to this research to in this field with this compound. And of course, I would like to thank all of you for your attention. So thank you, everybody.
All right, thank you very much, Maria, for a very interesting and exciting presentation of this compound. So the paper is now open for discussion. If anyone is interested in posting questions to Maria, please do so by using the raise hand option in your screen, and I will give you the floor to pose the question yourself. In case someone wants to make a question and have no microphone available, please send me a message through the chat, and I will write down your question, and I will transfer the questions to the speaker. Okay, so while we wait the question, let me remind everyone that this seminar is being recorded, and the recording will be available at the CyberNet website in the next few days, couple of weeks. And this CyberNet webinar series will resume after the Christmas break, and if anyone is interested in participating, please send us a message, an email to either Teresa Iglesias or myself, and I will try to do the best to accommodate a proper slot for you to present in this format. Okay, so any questions? I don't see any raised hands here. I don't see any message coming through the chat either.
It's clearly.
Yeah.
It's clear.
All right. Okay, so I will make a question myself, which is more of a general type of questions, Maria, if you don't mind. The question is regarding what you can share with us regarding the pharmacokinetics of this particular compound. I heard from the last couple of slides that the compound is already in clinical trial. So I guess it's all the regulatory preclinical toxicology has been done. And
yes.
But how, you know, what you can share with us in that?
We didn't develop any pharmacokinetics assays at in vitro levels, but this compound is generated and is prepared by a pharmaceutical company, and they have developed all the proofs that they need to assert the pharmacokinetics and the toxicity and the safety of this compound. And this is formulated now for clinical trials. So this compound we think is safety at very expert levels, and we think in clinical trial, the phase 1, it's now the moment to test that it's safety for humans, and we hope everything is okay, because in vitro proofs were okay in the pharmaceutical company. So we hope that.
Okay. So I see in some of your slides that you have used in the in vivo models, you have used IP administration in some cases and oral administration in some other cases.
Yeah.
Have you observed any major difference in this between these two administration routes? Is one better than the other, or because the concentrations are not that different in both, right? You use 10 mg per kilo in case of the IP, and around 20 when you use oral administration.
Yeah, always oral administration is worse for pharmacokinetics. So because of that, we decided to increase the dose, and because in the first oral treatment, we showed that the best concentration was 20 milligram per kilo, but we test 10 too, but with 10 milligram per kilo in the first oral treatment, we didn't get enough results or enough signification. So for oral treatment, we decided to employ from that moment 20 milligram kilo. And by intraperitoneal treatment, we started with 10 because we think it was enough, because when we translate from very in vitro level to murine level, we decided, and it was okay at 10 milligram kilo, so we decided it was okay. We use both different mode of treatment. First, we decide to use intraperitoneal administration because it's easier and its response is most evident. But in order to obtain a more clinic approach, we decide to treat animals with oral administration, but the only thing we should change is to up the dose of the compound. But this was the reason.
Okay.
The reasons.
All right, thanks. Last question from my side is, do you have any idea, or have you determined the ratio between blood and brain in concentrations of the compound? Any idea of how much crossed the BBB?
We suppose it crossed the BBB because we have obtained results.
Sure.
But we don't know the range or the ratio. We didn't study that. Maybe the company have more studies that we have in our hands, but I'm not sure what about that. So.
All right. Okay. Thank you very much, Maria.
Thank you.
Any other questions from the audience? Don't be shy.
Everything's okay. It's clear.
Yeah, I don't see any raised hands, so let me go through the chat just in case. No, I don't see any questions there either. Okay, so let me give you the last chance to pose a question. So this is the last call for questions, if anyone is interested. Otherwise, I guess that if anyone is interested in getting in touch with Maria afterwards, you can do it through email or phone.
Yes, of course. Of course.
Sure. All right.
It will be a pleasure to give you any information.
Okay. And all right, so if there are no more questions, then I think it's time to close the session here. But not before thanking again Maria for her willingness to share her work with us.
Thank you.
And thank you everyone for attending. And I wish you all a very nice holiday season that is approaching, and wonderful end of the year and the beginning of 2024, which hopefully will be very productive and most importantly, healthy to everyone. All right, so thank you everyone. Thank you, Maria. I'll see you next time. Bye-bye.
See you next. Bye.